So, hi everyone, welcome to this new session. So today it's our great pleasure to have two speakers. So the first speaker is Cheng Lyu, and so he worked previously at Rockefeller with Maimon and Gabby Maimon and Larry Abbott, and he will present that work. He's now at Stanford University. And so he will talk about how the fly's brain do math to compute the flight direction, but he will give, of course, much more detail about it. And the second talk is from Logan Shariker, who was who did his PhD and postdoc at Courant with Lei Sang-Young, and he worked with Bob Shapley too. And it's about network of E and I neurons and to model visual cortex. So today, these are really very applied math because they very deal with real applications. And so it's my pleasure to have them today for BDD. All right.
All right, thank you, Ben, for the invitation. My name is Cheng. I'll talk about how brains add vectors today. And more explicitly, I'll try to talk about how brains perform these three basic operations of vector addition, like vector scaling, rotation, and addition. Although I study fly, I always keep thinking about how any brains, or especially how mammalian brains, our brains could do math. So let's start with listening to the spike train of head direction cell in a rat. Did you hear the sound? The audio okay?
No, we didn't hear the sound.
Okay. I think given the limited of times, you hear a lot of spectrums when the rat's head is pointed to this upper left direction. I think trying to fix this may take a few minutes. But I don't have much videos in my talk. So here is the tuning curve of the cells we just recorded, just shown. And you can see this cell has a sharp tuning curve at this orientation, which corresponds to the upper left of the animal's environment. And so this high direction cell, since it's discovered, has many additional features that kind of remain unclear or only have models for. And this is just to show that there are supposedly many high direction cells that could tile the entire angular space. But occasionally, or sometimes, you can find cells from mouse brain or rat brain that has a tuning curve to a direction, but with a much broader tuning curve. And sometimes this tuning curve fit well with a sinusoid, which is shown in this dashed line here. And so it's been curious what are the function of the shape of this high direction cell tuning curve. And more recently, actually, early this year, it's been published a paper by Ruben Portugues lab where they record in the hindbrain of liverfish, and it's also high direction cell like activity, but you can see the somas are spatialized are localized in a very nice way that you can actually see activity bump in these cells. This is when the animal is moving, and you can see the activity bump changes, indicating the animal thinks he's changing directions. And if you look at the individual cell from this population, this is the soma. It projects to another brain structure called the interpedunculus nucleus. But interestingly, the dendrites and the axons are separated and quite largely separated, almost to the half the extent of this structure. So what are the function of this large separation between dendrins and axons of high direction cells? And lastly but not least, it's been known that high direction cells, if you record it more often than not, you will see like conjunctive tuning to other spatial information, such as the animal's location, the animal's speed, etc. So what are the functions of this pervasive conjunctive tuning in high direction cell? So that's from the mouse and the rats. And so the purpose of studying flies also to add insight onto this question marks, although there are many models already trying to characterize the function of this, but hopefully after today, I can provide some experimental data from flies similar to this observations. Okay, so flies also have high direction cells, and it's first been shown by Johannes Sillig and Vivek Jarma in 2015, almost 8 years from now. Since the audio is now working, here's a virtual trajectory of the fly, and now this cell also respond crazily when the fly is headed toward the upper left direction. And the tuning cell is very similar from the one you see from the rat. So this is one cell. It's actually called, before moving on to the anatomy of these neurons, the reason this is virtual, like a cartoon-like video, is because we can't put electrode onto a freely moving fly. So the way we do it, as Ben mentioned earlier, is that we tether the fly to a plate and put the fly either on air cushion ball like here, and she can walk around, and we hooked up this bright bar in a closed loop with the rotation of the ball. So it's to mimic a landmark at infinity to the fly, kind of to simulate the high direction of the fly in reality. So we can record neural activities in flies, in walking flies, and also do similar recording in flying flies, also tethered. And this is one of the video that we collected when we record from flying flies. Also, we can add this visual information or other stimulus to the fly. So this is the stimulus I'll use quite a lot in today's talk. So back to the neurons. The high direction cells shown on the left are actually recordings from one of these blue neurons called EPG. So they tile almost all the ellipsoid body, this donut-like structure, and most of the peripheral bridge, this bicycle handler like brain structure. They are located in the center of the fly brain. So these are the population of these neurons, but if you look at each one of them, they only innervate one wedge of the ellipsoid body and goes up and send axonal output to one glomerulus of the peripheral bridge. So this neuron, this goes to the left, and if you record from this neuron, you will have a tuning curve shown previously. But the neighboring neuron have unique anatomical pattern that will go to the right bridge and left, right, left, right. So through this alternating pattern, if these neurons are active, what you will see if you image all the neuron activity of these EPG cells, you will see this activity bump, similar to what was shown before in the zebrafish brain, that you will see 3 bumps here, one from the ellipsoid body, and the 2 others from the same neuronal populations, but in the bridge. And of course, if all the neurons take turns of being active, like when the fly is turning around in space, what you will see under the microscope is this bump and rotate in a synchronous way, all indicating the heading direction, of the cell, of the fly, head or heading. So back to the title of this talk, how do brains add vectors? So this is one of the the key component of the vector computation. Another key component is the traveling direction. It's not trivial to distinguish hiding and traveling direction. For example, in humans, when we walk, we can walk in, look at a different direction when we walk. And this is an ant going home backwards, shown by the study of Matt Willinger, that they sometimes, when the food is super heavy, they walk home backwards. And there are many behavioral evidence indicating that throughout this process, this ant still have a sense of their traveling direction, which is different than their heading, which is like 180 degree apart in this case. And in flying animals, here is some blue mannequins. The males are courtshiping the females, and you can see the gaze at the female during each dance and traveling to the side. So being able to distinguish hiding and traveling direction is very important in spatial navigation. But so far, maybe not so far, maybe up to 2 years ago, 1 year ago, only high direction cells has been shown, and it has been shown in almost all the animals. You can see there are many different animal species have this high direction cell, but the traveling direction cell hasn't been shown until like a year ago. And since the focus of this talk is how do brains add vectors, I'll quickly show you the evidence we think for this traveling direction and focus on how the brains add vectors. In short, these traveling direction signals in the fruit flies, they locate in this fan-shaped body, this middle layer of the fan-shaped body, in two groups of neurons at least. One is called H dot B, one is called PFRs. So we'll only talk about H dot B's in this talk. They also have a bump of lactic activity moving left and right along the fan-shaped body. So these three structures are closely located, and they all belong to the same structure in the fly brain called central complex. So intuitively, it should be very straightforward if we record this neural activity and also simultaneously record the traveling direction in, for example, a walking fly. We should be able to correlate these two signals and show if it's a traveling direction signal. But unfortunately, the traveling direction of a fly when it's walking is changing so fast. Like when it jitters a little bit to the left in maybe a sub 100 milliseconds, the traveling direction will change dramatically. And the GCamp indicator from that we use or exist do not respond in such a fast way. So to get things working, we switched to recording from flying flies. But the problem in flying flies is that it's tethered. They're not actually traveling to any places, to any direction. So the trick we use is to not record the actual movement of the fly, but to use VR to show the fly which direction you are traveling, kind of like the virtual reality we're using now in humans. So the way we do it is to give the fly this optic flow thing, that's this dim dots on the lower part of the arena that's surrounding the fly. So when we move these dim dots collectively in this manner, it's simulating what you will feel like when you are moving forward. And when you're moving forward, your head direction and traveling direction are aligned. And this is what we observed in these two groups of neurons. We simultaneously record activity from these two groups of neurons. Now the x-axis is time, and between the two dashed lines are the moments where this optic flow is going. And the y-axis is the unwrapped, the linearized this brain structure. So the top row is EPG that simulates indicates hiding direction. So now the flashing hiding direction is to the edge of this y-axis, and the traveling direction from H dot B, this bump, also goes to the edge. And we can estimate the phase from these two patterns, and they show that when the stimulus is on, both phases aligned, indicating the hiding and traveling direction aligned. And across a population of 13 flies, this is also true. So this is just one direction. Let's test more other directions. Another direction is to simulate the fly going backward. And when we display this movie, what we saw is that now the head direction, it keeps changing, but that's why we record this head direction simultaneously. So the head direction, it goes to the edge, and the traveling direction, it locates to the center, and they are on average 180 degrees apart from each other. And we also did this recording in other 4 directions, so in total 6 directions, and you can see the separation of these 2 bumps actually changes in a graded manner, and on average they form a very linear correlation with the simulated egocentric traveling direction. So egocentric here is a word, means this traveling direction is with reference to the fly's body, like going front, back. And there's another word called allocentric direction, traveling direction, with reference to the world, like going south, north, west, or east. We'll need to distinguish these two in the rest of the talk. So to summarize this first part, now there are these 2 bumps in the fly brain. One, the blue bump tracks the fly's heading direction. The pink or red bump tracks the fly's traveling direction. So when the fly is flying around, if they move forward, both bumps align. And when the fly is blown backwards, the traveling bump switch 180 degrees, whereas the blue bump remains still. Here's another example. So we think this is true. So we show that it's the case in flying flies, and we also have a lot of data showing this is qualitatively or semi-quantitatively true in walking flies. And for more, if you want, we think, so now so we think this pink bump is to represent hopefully the abstract traveling direction of the animal, no matter it's flying or walking. So how do the fly brain build this allocentric traveling direction signal? Because the x-axis now correlates with the southeast northwest. How do the fly brain build this allocentric traveling direction? So I think this process can be summarized as a vector computation process. So first, let me go through the algorithm we think that's going on, why this is a vector computation process. So here are the 2 angles, the same abstract, the same angle, but reference to different. The one on the left is egocentric, and the one on the right is allocentric. So the difference between these 2 angles are the fly's heading direction. And so you add ego traveling with heading, you got allo traveling. Okay, so what we want is allocentric traveling direction, but egocentric traveling direction is seemingly more easier to compute. So there are actually there are recordings from neural activities in insects, previously from bumblebees, also shown in this work, that we have like neural activity whose activity correlates nicely with the length of this egocentric traveling vector projected onto this positive 45 degree axis. I know it's very arbitrary, but in short, is we have neural activity that correlates with this length L, which is the projection of traveling direction. And we have different neural activities from different populations that correlates with different length L, which is the projection of this egocentric traveling direction onto these 4 different axes. So one way the fly can reconstruct this green arrow is to use those neural activity, those gray arrows, and sum it up, and then you can get the green arrow. And this is the step we think is a vector addition. And once you have this green arrow, you just rotate it by the amount of h, and then you got allocentric traveling direction. And this is what we call, we think it's a coordinate transformation. Now you can switch from egocentric to allocentric. So this is done in 2 steps. And what's actually done in the fly brain is that they combine these 2 steps into 1 step. So the reason this is a green arrow is in egocentric reference frame is because these 4 axes they use to compute vectors are referenced to their body. So if these axes are directly referenced to the allocentric world, then the vectors they got, the summed vector automatically becomes reference to the allocentric world. So this is what we think is going on in the fly brain. And throughout the rest of the talk, I'll only talk about data that we think is reflecting this process. So first of all, how do neurons like represent vectors? So actually, I think a trick that's been used is called a phasor representation of this vector. So every 2-dimensional vectors you can have this length and angle, and you can also represent it in a sinusoidal way, where the length, the angle equals the phase of the sinusoids, and the length of the vector equals the amplitude. The amplitude is very important, the amplitude of the sinusoid. And if you have another vector, you just have another sinusoid. And if you want to add these 2 vectors together, you just add these 2 sinusoids together. So since these 2 sinusoids are of the same frequency, the added sum, the sum is also a sinusoid of the same frequency. So this is a mathematically true. And this is actually a idea that actually been put forward in many models, theoretical models that use to calculate, to understand or to explain many spatial navigation tasks. And the one thing I forgot to mention is that in neurons, it's actually much easier to represent the things on the right than the things on the left, because to represent one sinusoid, you can use one group of neurons, say this one drawn on the bottom. And so each subgroup of this neuron can represent each beam of the sinusoids, and the height of each beam in this sinusoids can be represented by the activity of this subgroup of neurons. So altogether, this group of neurons with their activity, they can use to have a 1-to-1 mapping between this purple sinusoid. And you can have different groups of neurons to represent different sinusoids, and add them together, you can achieve vector computation in the fly brain. So that's, I think, the take-home message if you don't understand anything I say from now on. Okay, now the biological evidence for it. So first, we need more copies of hiding direction, or more copies of the sinusoids. So that's the theoretical and computational paper, including one also have from Barbara Webb and Stone and Nat Hans that also have experimental data in bees. Okay, so the more copies of the hiding of these bumps. So one way to look at them is in the output region of this EPG cells, which is the hiding direction cells in the fly brain that we talked about earlier. So in the ellipsoid body, it's dendrites, and in the bridge, it's output. So they have 2 output, one on the left, one on the right. They are all the same, first order. So if we look at the output of these EPG cells, we found these 2 other groups of neurons that they both innervate the left and right bridge and send their output to the fan-shaped body. And these 2 are called PFND and PFNV cells. It's a lot of names, but I'll always color code them to make things easier. So when we simultaneously record neural activities from EPG cells and PFNV cells, for example, in the bridge, we saw these 2 bumps of activity in the EPG cells and also 2 bumps of activity in the PFNV cells that are moving along the bridge synchronously, and they also have overlapping bump of activity, suggesting this PFN cells more or less is receiving the EPG cells input. Okay, so that's now we have 4 additional bumps that all track the heading direction of the fly. As it skips the data from PFND cells. So, but to compare, if we look at the shape of this heading direction cell of the neural activities, for example, if we look at EPG cells, they have a very narrow tuning curve. This is consistent with the data we got from single EPG cells from electrophysiology recordings, where when the heading direction of the fly changed a little bit, the EPG cells will lose its activity. It's very sharply tuned to their preferred direction. And here, if we image them all together, you can see that they are also, the shape is very narrow. But compared to this narrowly distributed shape, here are the PFNV cells. They have a much broader, wider shape. And if we try to fit this shape to a sinusoid, they can actually fit quite nicely. So this is at least one piece of evidence that this orange, and it's the same, it's true for this brown bumps as well. So it's now we have this 4 wider bump, that's a sinusoidal-like shape, and 2 narrow bump in EPG cells. They all the 6 bumps as they track the fly heading direction when the fly moves around in the world. Okay. Now we actually met the 2 criteria for vector computation. One is we have more components, more heading bumps. The second is that these bumps are sinusoidal shape. Okay, a third requirement for vector computation is the following. So these 4 bumps, because their phase all pointing to the heading direction of the fly, if you put them together, they have the peak point to the same direction. But what we need is this is what they look like in using a Phasor diagram in 2D space. They all pointing to the same direction, the vectors they represent. What we need are these vectors to point to different directions. And this in return will require these bumps to have peaks, to have their phase also separated. And this is the third requirement, that we need the phase shift between these different sinusoidal bumps. And this can be achieved when through the anatomy of these neurons, that they project from their dendritic area towards their axonal output area, from the bridge to the fan-shaped body. So let's focus on one of these cells, these 2 pairs, these 2 PFNB cells that they innervate the center of each side of the bridge. But when they propagate to the fan-shaped body, the one on the right bridge shifted a little bit to the left, and the one on the left bridge shifted a little bit to the right. And if you say the whole layer is like 360 degree, because the bump sweeps through the whole layer when the fly changes traveling direction 360 degree, then this shift, we can quantify the amount of this anatomical shift, and it's roughly 1/8 the width of the whole layer, and which corresponds to 45 degrees left or minus. And I just want to give a highlight to this work from Tanya Wolf and Jerry Rubin, that in 2014, which is almost 10 years from now, they used light microscopy to sparsely labeling these neurons, and they actually have this qualitatively, they have this conclusion that this PFN neurons, when they project from bridge to the fan-shaped body, they have some anatomical shift. And more recently, from this hemibrain electron microscopy dataset, and we can more accurately quantify this effect. I'll show the quantification results later, but this is a EM picture, reconstructing PFNV neurons. PFNV neurons, yes, from the bridge to the fan shaped body. You can see they shifted a little bit to the right. So let's say this is 45 degree plus or minus, but that's not the end of the story. So this means the sinusoids, the hiding bump, when they propagate from the bridge to the fan shaped body through PFNV neuron, they have this anatomical shift of 45 degree. But they need to actually travel to the axonal output of this H.B neurons, which is where we see traveling direction in the axonal output of this H.B neurons. So to travel from the dendritic field to the axonal output of the RAN neurons, there is another half layer switch, which corresponds to 180-degree switch. So the sinusoids from the bridge to the tip of the H.B neurons actually have a combined minus 135-degree shift. So that's from the left bridge of PFNV cells, and from the right bridge PFNV cells, this shift is positive 135 degree. And for PFND cells, things are very similar. They project to the fan shaped body, they have this 45-degree shift, they connect to H.B neurons, but they connect very weakly to the dendrites of this of this H.B neuron, of the RAN neurons. But much more intensively, they connect directly to the output of the H.B neurons, which is rarely observed in the orange PFNV neurons. Because if you connect to the output versus the dendrites of the RAN neurons is have the opposite effect because they are 180 degree apart. So it's kind of like winner takes all, and we can, for simplicity, we can just think about this axonal direction connection. So in this case, there's no additional 135-degree shift. So the PFND cells from the right bridge have 45-degree shift in total, and from the left bridge have minus 45-degree shift in total. Yeah, so this is a parallel work published with Richard Wilson's lab, and where they also show similar ideas of how this anatomical shift plays a role in vector computation by Richard Wilson and Cheng Lyu. Okay, so in sum, now these 4 sinusoids that are phase aligned in the bridge, all tracking the EPG, the heading direction of the flight, as shown on the right. When they propagate to the fan shaped body, to the tip of this H.B neurons, they each inherit a different shift. The left bridge PFND cells have minus 45-degree shift. I rotated the angle 45 degree. The right bridge positive 45 degree and positive 135 and negative 135. So this is what we think how these neurons achieve this phase shift. And this is just to show it's a great year for people working in flight, where this EM data is quite accurate, that we're just using the synapses detected from this EM data between different groups of neurons, and we calculated this anatomical shift. It turns out very close to a perpendicular orthogonal axis. So now we have 4 sinusoids that pointing to the right direction in the fan shaped body. And the last step is to have this sinusoid have the vectors that they represented to have different lengths, and not just changing length, but also changing length in a very specific manner, because when the flight changed her traveling direction, these vectors will change their length in a sinusoidal way, because it's a projection relationship. And So this in turn requires these sinusoids to change their amplitude. So that's the last thing we try to observe in experiments. That is that whether these four sinusoids will change their amplitude in a sinusoidal way. So there are two sinusoids here. So we image the activity of these neurons in the bridge. That's where we can separate them. So when we simulate the fly going backwards, the PFND cells have very small amplitude of their bump, whereas PFNV cells have very large bump. And the same groups of flies, when we change the traveling direction, stimulate the traveling direction to the front, you can see the PFND bump increase their sinusoid, increase their amplitudes, and PFNV bump decrease their amplitudes. And this is true for different traveling directions simulated, and this bump become asymmetric, asymmetrically have different amplitude. So we can quantify the amplitude of each sinusoid in this picture. And what we got is, for example, the left bridge PFND cells. What we got is this mean tuning, this tuning curve that's close to a sphated sinusoid, with the peak of this fading of the sphated sinusoid pointing to the upper left. So this upper left is actually their maximum responding direction. And there's another upper left for this PFND, left bridge PFND cells, which is their anatomical projection shift. So the fact that these two angles, they match each other means that the PFND cell from the left bridge, if they are advocating for this upper left direction, they should respond more strongly to this upper left direction. This is also a prediction from the model. And the same is true for the right bridge PFND cells and left right bridge PFNB cell, where you can see their maximum response to the front, to the back left and back right. Just as a control, this EPG cells that signals the hiding direction of the fly do not change their amplitude when we change the traveling direction of the fly. Okay, so here we are. We have all the components, and let me summarize this in a video with no sound. The video has no sound. So here's the fly traveling in space. For simplicity, we just in this cartoon we showed the fly's head. It's always pointing to the up. So the hiding direction of the fly never changed, but she's constantly changing her traveling direction, represented by this red bump sweeping through the fancy body. So how does this red bump calculate it? First of all, the same traveling direction is not projected onto this front, back, left, right axis, which we human used to. They actually projected onto its four other axes, also orthogonal axis. So the reason I say this is because some data I didn't show in this talk for the sake of time, that we have these four different groups of neurons whose activity correlates with the length of projection onto each axis. And actually they are anti-correlated. So we think it may show some quite interesting feature why this regulation needs to be to be done in a disinhibitory manner. But nevertheless, this is the traveling direction of the fly projected onto these four different axes. So now this neurons only signal not a vector, but one-dimensional variable. So the step that they become vector is where they combine with EPG, this hiding bump, and they combine it in with the help of other neurons, which I didn't talk about, that they become sinusoidal shaped bump in this PFN cells. And there are four bumps of this PFN cells, and they conjunctively they signal the flight's heading direction by the phase of the bump and signal the flight's speed onto one of these four axes by the amplitude of the sinusoid. And this is the step where vector scaling is achieved in the flight brain. And now these bumps all pointing to the same direction. The step where they achieve different shifts is through this anatomical projection from the bridge to the fan-shaped body. And this is the step where the vector rotation is implemented. So here in the fan-shaped body, these four sinusoids, they all they are all summed by the same group of neurons, which is this red neuron called H.B neurons. And the sum is also a sinusoid, and with the peak corresponds to this red dot, which is the flight's traveling direction. And this is the step where vector summation is achieved. And so at the last slide, let me go back to this data from the vertebrates. So now we have similar activity of neurons compared to this data from vertebrates. So now what are their functions? It's not saying that these are the function of these features, but just to give some insight. So this broad tuning curve of the heading direction itself could be used to explicitly represent 2D vectors. And this anatomical shift between dendrites and axons shown in the vertebrate 180 degree as an example, and actually in flight, there are many different levels of shift. And we think the function of this could be to serve as to rotate the 2D vector. And lastly, this conjunctive tuning of heading direction with other signals could be used to as a coordinate transformation to transform the other signal into the reference where the heading direction resides. So with that, I end my talk and thank my PhD mentor, Gabby Mayman, and my collaborator and also my co-mentor, Larry Abbott from Columbia. And I want to thank everyone from the Mayman Lab where we work together to crack this problem. And here are the flights we received for this project. And here are the fundings. Thank you. That's it.